lecture. And thank you very much, sir.
Thank you so much for having me and giving me the opportunity. Thank you so much.
Gurjesh ma'am, our second speaker with us. Gurjesh ma'am.
Yusuf sir, are you there? Yusuf sir, are you there?
Yeah, I'm here. Can you hear me?
Yes,
Now
sir.
I
Can
would
you-
like to
Harsha ma'am,
invite
please
our
continue.
next honorable speaker, Dr. Yusuf Akhtar, Assistant Professor, Department of Biotechnology, School of Life Sciences, Baba Saheb Bhimrao Ambedkar University, Lucknow. He has a rich teaching experience, more than 13 years in various eminent institutions. In his research field of biotechnology, he has specialized in protein structure and infection biology, and has published 107 papers in Scopus 1 and 2 journals. He has conducted 8 workshops and training programs and has been invited to 25 talks. He has also received Research and Teaching Excellence Award from Baba Saheb Bhimrao Ambedkar University, Lucknow. He also received the Sentinel of Science Prizes by Publon for top peer reviewer and editorial contribution worldwide in 2016. He also received DAAD Long-Term Fellowship in 2007, EMBO Long-Term Fellowship in 2010, and many more fellowships. He also associate editor of scientific reports, Nature Research, Frontier in Microbiology, etc. He also associated as visiting faculty in University Technology Malaysia, Zoha Baru. He has guided 9 students with their PhD and more than 50 PG students with their research work. Today, Dr. Yusuf Akhtar is going to share his vast knowledge on post-COVID-19 trends in drug discovery against infectious disease. I would like to invite him for the lecture. Over to you, sir.
Okay, thank you very much for your generous invitation and now generous introduction. So I will not waste much time, and I would like to share my slides and start talking about the topic on which we are going to have this lecture for next. Yeah. Screen is visible. Okay, so I think my screen is.
Excuse me, sir. Sorry to interrupt you. Your voice is too low. Can you increase your volume of your system?
Okay, wait a minute.
It's not audible.
Hello, could you hear me now?
Yes, sir.
Okay, so like the earlier speaker, he has covered very diverse range of technologies and therapeutic and medical interventions advancement due to this pandemic, whose 3 waves uh, around the world have made havoc. So I will in my lecture I will more focus on drug discovery portion, because that is my area of expertise. So like the title says, post COVID-19 trends in drug discovery against infectious diseases. So we will try to cover several topics, but the whole discussion will be around the drug discovery process and how it happens, and what are the basics of drug discovery, and with a special reference to COVID-19 and other infectious agents. Okay, so let's come to first, means there are diseases. If we broadly divide diseases based on whether it is caused by infectious agent, or whether it is an aberrant state of in an individual's physiology, but without any infectious agent. Okay, so infectious disease versus metabolic diseases, like caused by microbial infection. So at one side, infectious disease is usually caused by a microorganism. That could be viruses, that could be bacteria, or fungi, or protozoans, or worms. Okay, and the other type of diseases like metabolic diseases are there, like diabetes, like hypertension, that don't involve any infectious agent. Okay. So these have modes of transmission and spread from one person to another person. So these infectious diseases, these must have mode of transmission. So like there are there are food-borne infections, there are water-borne infections, there are vector-borne infections. Vector-borne, like in northern Indian states now, there is a lot of dengue fever. There is, in these months, November, December, you will face a lot of dengue fever, a lot of chikungunya, and other type of flus. So these are like these are spread by vector. Vector is what? Vector is some organism which is intermediary host. Like in this case, in the case of dengue fever, it is Aedes aegypti virus. Sorry, Aedes aegypti mosquito. So mosquito basically carry, when it bites one infected person, it will it will with the blood, it will take also, it will take also the virus with the with its bite, and when it will bite again to a healthy person, it will transmit it. Similarly, like there are like amoebiasis, one type of protozoan which causes diarrhea. Okay, so that is by infected food or infected water that happens. Similarly, there are other diseases like other viral diseases and other infections, like sexually transmitted diseases. So that is caused by that is spread by one-to-one contact. Okay, so then blood transfusion also, like if an infected blood is transfused to a healthy person, then he will become infected. Then there are other ways of transmission, like from mother to the newborn. So these are infectious diseases. So these happen like that. But in contrast, metabolic diseases, in metabolic diseases, there is a aberrant function of the body, some vital organ, or some glands, or some any particular organ which will not function properly, and then it can happen. Or also it can happen due to aging and senescence. When people grow older, then a lot of cells die, and a lot of tissues become weak. Or also it can happen by malnutrition, means uh, when people will not eat proper food. Okay, so now, like I said, I am a person of infection biology, so I will focus on the topic. how drug target selection against a microbial pathogen is being done. So in a simple, because the audience are of diverse nature, so let's try to understand how it happens, how we select a target, how we design a drug. So what we will do, basically, we there when we analyze what is best target against which we can design a drug. So what we will do, basically, we look at the microbial pathogens component or host component. So so primarily it can be targeted to microbial pathogen components. Means, let's say, if we design a molecule, if we design a drug which goes and blocks the microbial pathogens some component, and what happens then, so this component should be such that that microbe will not divide and that microbe will die. Okay, so it can happen like that. In some special cases, we can also target host components, because when microbial pathogen, either that is bacteria or that is virus or that is fungal pathogen or worm or anything, when it
Excuse me,
infests
sir.
or in, yeah, could you
सर
hear me?
प्लीज आप अपनी स्क्रीन जो है एक बार शेयरिंग हटा के फिर से स्टार्ट कीजिए शेयरिंग, बिकॉज हम लोगों को फर्स्ट स्क्रीन ही दिख रही है सर.
Okay, wait a minute. Now could you see?
यस सर, बट आई थिंक आपने स्लाइड शो शेयर नहीं किया है. अभी भी हम लोगों को जो पावर पॉइंट है बस उसमें थर्ड स्लाइड दिख रही है, तो बेटर यू
तो
शेयर
जब
योर कंप्लीट स्क्रीन.
No, I have shared before, and when we will share, so in these programs, this is a problem. So if I will slide this one by one, then I think it will work. Otherwise,
Okay.
again it will stick.
ओके सर,
Okay.
ओके सर. यस.
Okay. So I think this is good to see, है ना? You can see that, na? Third slide.
यस सर.
Okay, wonderful. So now, like I was talking, either we will block some component of that bacteria or virus, or in rare cases we can block the host components. So but when we will block the host component, there is chance of side effects always. Even when we will block the microbial component, then also there will be off target effects, off targets or side effects. So that's why drug discovery is very slow process, and it needs a lot of evaluation, and it only completes when all the biosafety, all the clinical safety experiments and trials performed carefully, and all the data, all the reports are checked by the authority, or the um, or every country has means these food and drug authority or food and drug administration type of bodies. Like in India also, there is there is uh, an agency which basically, like in America there is FDA. Here in India also there is agency which look at it, and then only the license is issued. So it is a careful work anyway. So where the drug targets are there, where the host and the pathogen, they interact basically, because if there are host components are also involved, which allows the pathogen, microbial pathogen or infectious agent, to enter the host cells or host tissues, and basically these microbial pathogens or infectious agents or parasites, they feeds upon the host resources and host space. So there may be, in some cases, we can target also host components. Okay. So now the slide is changed. Slide is changed or not?
Yes, sir.
Now you are okay. Wonderful. Okay, wonderful. So if we, so let's say if we look at what kind of pathogens component it could be targeted. So like I have given here one example of when we design drugs against a bacteria. So how these bacterial components could be targeted. So you see there is a kind of anatomy of a bacterial cell. You can see there is cell wall, you can see there is cytoplasm, and there are ribosomes, there is DNA in between. Okay. So these all parts, these all components are required for the functioning of this bacteria. And then if everything will work properly, so it is also like living organism, animals and higher organisms. So if these things work in a in a proper manner, then only bacteria will survive and bacteria will divide. Okay, so there are these number of drugs. For example, cell wall. So cell wall of the bacteria is previously targeted by these so many antibiotics. You can see beta lactam group antibiotics, which very popular beta lactam antibiotic is penicillin or ampicillin, which 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 is an old and probably the first antibiotic discovered. Okay. Then there are cephalosporins, there are monobactams, there are carbapenems. These all are derivatives of beta lactams. Then there are glycopeptides like vancomycin and bacteriacin. So there similarly, like here cell wall is targeted. Then in another case, like plasma membrane is targeted, like polymyxins. By polymyxin B, cholestyne. Then there are lipopeptide which also target the membrane of the bacteria. Then there is daptomycin. Similarly, ribosomes are also blocked by many known antibiotics like aminoglycosides, tetracycline, and microlides, etc., etc. Okay. Then there are metabolic pathways. So metabolic pathways in any cell, any living cell, it, there are several biological catalysts, like we call them enzymes. So what enzymes do? At a particular time in a living cell, there are hundreds of compounds are converted into one compound to another compound. And means because we are let's say we are eating food has nutrition like carbohydrate, like protein, uh, like uh, fat, other things. So it will be digested, and when it will be digested, it will be assimilated and absorbed by the intestine, and it will go to the blood, and blood will take it to all the tissues, and where there all tissues are made up of cells. So cells will take this nutrition and they will continuously oxidizing it and converting it into another component, another metabolite. Similarly, there are a lot of biosynthesis is going on, there are a lot of degradation is going on. So a lot of chemicals are converted into one form to another form. So for that we need enzymes, because otherwise if the process is not catalyzed by enzymes, it will be very slow and very inefficient. So those enzymes could be targeted. Those enzymes could be targeted by drugs and could be blocked. So now the point is that, let's say we want to target any bacterial component. So what that component should look like, what basically that component, if we want an efficient drug, then that component should be unique if we compare with the human, because if it will also target the human component or human protein or human cell structure, then what will happen? It will create a very bad side effect, and then it is not a good drug. Okay, so that's why when scientists analyze what they try to look, they basically look a unique component in bacteria which should not be present in the human cells. Okay, so like these drugs you can see on the screen, these are successful antibiotics. Those are discovered in last maybe 70 years, and these all have, okay, those can have side effects, but not huge side effect. Like if somebody is suffering from a bacterial infection, he will take a course of antibiotic for 7 days or for 14 days or for 5 days, depend upon what kind of antibiotic you are taking, and that time period is already optimized, that in this time period that bacteria is completely killed from the system and removed from the system. So those are, and then you took for that period, and then you are fine, and if there is a small side effect, then you recovered. Or doctors also prescribe like vitamins and other drugs like proton pump inhibitor with the antibiotics, so that the side effects will be subsided. Okay, so let's go to so I gave the example how drug targets are is being selected. So now let's focus on more on this virus, because that is the topic of this your seminar. So this SARS coronavirus, uh, SARS-CoV-2 or SARS coronavirus 2. So basically, this SARS is a family of coronaviruses uh, which usually infect animals, but in last 20 years there are other coronaviruses which started infecting humans. So there was one in one was SARS. Its name was SARS coronavirus. So SARS uh, and COV, not -2 like this one. So that was earlier, which was modified, and then this new one was discovered in 2019. And there was another one which was called as MERS, MERS, MERS. That was also coronavirus, and that was spreaded in Middle Eastern countries. So anyway, so how this coronavirus look like, if we look at it and little bit go into detail, how what components, uh, it is made up of what components basically. So if we look at it, then virus, how the coronavirus look from inside. Okay, so you can see there is a outer layer. So in all viruses we know there is a outer layer which is made up of usually proteins, and then there are enveloped viruses. So enveloped viruses have one more lipid layer outside the outside the protein, and inside the protein coating there is nucleic acid, where all the information for its life, for its all the genes, these are present in that nucleic acid which is in the core. Okay, so like RNA, in case of coronavirus is a RNA virus. So there is a RNA genome inside the protein code. Okay, so there are a few number of proteins it is made up of, like spike protein. So its name corona means, in Latin corona means sun basically. So sun, why they started calling it like a sun. Corona means sun. So because of there is a protein which is called as a spike protein. So if you think of sun, how you depict or how you paint the figure of sun, basically you make a sphere or you make a circle, and then you show some spikes, the rays coming out of the sun. So that's why people who discovered very first time this virus, because you can see there is a spike protein which is coming out like a sun. So they named it coronavirus. Okay. So there is spike protein, and there are other proteins like E protein, membrane protein, M, and there is membrane. There is lipid membrane outside. So there is another depiction on the right side where you see nucleic acid, and then the capsid of the virus means protein coat, and then outside blue, within blue you can see the lipid coat, because this is an enveloped virus. So enveloped viruses have one lipid coat outside. Okay. So this looked like this, means you can see its anatomy. So like I said, when we target, when we try to look that if I will make a drug, I will make against any component of the pathogen or any component of the host which is involved during the disease, during the infection. When infection will establish, then that component will be involved. Okay. So here you can see a chart where you can see how coronavirus enters the human cells. So SARS coronavirus 2, it enters inside the human cell using a receptor, and the receptor is called ACE receptor, angiotensin converting enzyme 2, ACE2. So this ACE receptor basically is there on the cells for a different purpose, and that is related to hypertension and cardiovascular function, because angiotensin converting enzyme, that is related to that. But this virus uses the same receptor. This is quite interesting. Okay. So what it does, and there is a co-receptor, it is called as TMPRSS, TMPRSS. So what happens if on any cell these two receptors are not there, then basically this virus cannot enter that cell. And like other viruses, all viruses, if they don't get their host cell, they will not divide. They are like non-living particle. So what happens? It get attached to this receptor, and then it enters. And after entering, it make all the machinery. It make its all machinery in many copies, and it uses the host cell to make that machinery. So there is basic biological processes like transcription, translation, etc., with which it will make multiple copies of its genome, replication, transcription, and translation, and then RNA molecules, and then after that proteins, and then it will package itself. You can see in the end. So it will use all the host cell organelles, like it host cell Golgi body, it uses host cell endoplasmic reticulum, and RNA polymerase of host it will use, and then it will make all the machinery, and then it will make multiple number of virus particles, and in the end it will be released, either it will be released or it will burst the cells. So in the end the cell will die. So let's say it, so and it will then, means one virus came and it will make many copies of its own, and it will burst the cell and it will infect further cells. Ultimately the tissue, like in the case of SARS coronavirus 2, it is the first, it will enter in your nasal passage or mouth or from eyes, and then it will go into, it will take its path to the lungs, and there it will colonize and it will divide in the cells, and it will make multiple copies, and then it will infect, then those copies will infect further tissue. Okay, so now if, like we said, so virus based anti coronavirus drug discovery we can do, or we can do host based anti coronavirus drug discovery. So whatever we do, it should follow what? It should stop fusion of virus to the host cell, either it should stop replication of viral genome, or it should disrupt the assembly of the new virus particles, or it should stop the release of new virus particles after burst of the cell or after release of the cell. So for SARS coronavirus, so these are the checkpoints where, against which drug discovery can be done. Okay, but as virus use most of the host machinery to propagate itself, drug discovery against viruses are quite difficult, because we cannot target much the host component, because it will block, because because all the host component is evolved or made for another thing. For example, the ACE receptor is made for another thing. It is made to control blood pressure. But what is happening here? It is opportunistically used by the virus to enter the cells. Similarly, host RNA polymerase and host organelles are made to run your cells, your tissues, your body, but it is used by the virus as a parasite. Okay, so for SARS coronavirus 2, the receptor is ACE, which is present on all ciliated epithelial cells. For MERS, the earlier one, the receptor is different. That is Middle Eastern respiratory syndrome virus. So like this one is its older version was MERS, which was spread in Middle Eastern countries at some point, at like 12 years ago, and like 20 years ago there was this SARS coronavirus 1 was spread. Okay, so now we know that when the drug discovery will be made, then what components of either microbe or the human cell could be targeted. But targeting human cell is not, human cells component is not very preferable, like I said, because it is all human cell components are made to perform some function, some normal function, which is used by the virus opportunistically. Okay, so the drug discovery process, overall drug discovery process is very lengthy. Like you see in the cartoon, identify, if you want to do that, then you identify the disease, you isolate the protein, so it involves like 2 to 5 years, and then you find a drug effective against disease protein. That protein you blocks. Protein is what? Protein is either microbial component or host component, or you can say human component. Similarly, then preclinical trials like 1 to 3 years testing on the animal models. Similarly, and then formulation and scale up trial, and then human clinical trials. So this all will cost like 15 years at least. So drug discovery is very slow process, and there are a lot of, like earlier speaker was telling, that for drug discovery or for vaccine discovery in coronavirus, the regulatory bodies have speed up, they speeded up the process, they speeded up the means giving the license process, they speeded up much, so that the anticipated goals will be achieved. Okay, so drug discovery process and technologies. So there are target identification. So target identification could be done using, now the technology is advanced a lot. So genomics, gene expression profiling, a lot of bioinformatics and proteomics could be used. Then there is target validation. So target validation could be done using gene knockout in animal models, and then there are a lot of inhibition assays can be done, and then lead identification, high throughput screening using computational screening, and then combinatorial library could be used, and fragment based screening could be used. These are more finer techniques to do drug screening. Then there is lead optimization, like medicinal chemistry driven optimization, means what we have virtually or computationally screened, that lead will be synthesized in the lab. It means that compounds, the top, let's say top 3 compounds of from millions we have selected using computational tools, and then we synthesize it, and after synthesis, then real binding with the target in the experiment could be done. Later on the animals it could be tested. First what you do, first you make the disease model, a diseased animal model, and then you give your selected drug which came in your earlier analysis, and then you see whether the drug is able to treat the disease in animal or not, and then you go to next studies, like what pharmaceutical pharmacy people do. So pharmacodynamics, pharmacokinetics, etc. could be tested, and then in the end human trials can be done. Okay, so rational approach to drug discovery is, like I said, identify and validate the target, and then you have to produce that target using gene cloning and expression of that protein. And after expressing the protein, you do structural studies of that target that you can do by experimental ways and also with the help of computational ways, but in the end you have to test in experiment only to validate. So crystal structures or molecular targets of and inhibitor complexes could be tested. So whatever we have shortlisted in computational drug discovery, we have to test in experiment, and there what we do that either that host component or microbial component and our drug, we see whether it is able to bind or not, and if it is able to bind, then what is the mechanism of binding, and that binding is stable or not, and then synthesize modified lead compound accordingly. So the chemist, here the role of chemist is there. They modified that lead so that it will better bind, or it will not bind to the off targets, or it will not, then it will minimize the side effects, and then toxicity and pharmacokinetics testing, and then preclinical trials in animal models, like I said, and later on human trials. So there are many tools which could be used in the first in silico screening, like comparison of sequences can be done, and identification of targets, like I explained, that there are homology modeling, then systems biology approach for identifying targets. Then there are a lot of databases are there, which is used to curate and systematically arrange information about drug and drug targets. Then there is in silico screening, like ligand-based, receptor-based screening, etc. And then pharmacodynamics, pharmacogenomics databases, which assist basically all the genes and proteins where there is mutations, or there are lit- other information like uh, what is important, SNPs, mutations, etc. That is stored in the pharmacogenomics database, etc. Okay, so there are several processes like molecular docking, virtual high throughput screening, and then quantitative structure activity relationship. So these all are the methods which are used in this kind of drug discovery. So for example, we use molecular docking. So like I said before, like you see there is a ligand and there is a receptor. So this receptor is basically that human or microbial component which we want to block, and ligand is our inhibitor, which will go and it should go like a key, like lock and key, and it should go and it should block it. It should bind and it should block it, so that that component will stop functioning. And that component, if it will stop functioning, if it is a microbial component, then microbe should die. So we have to select such component. Okay, like I said. Okay, so there are these methods, means ligand based screening, etc. And then we calculate like binding energies. So binding energies means how strongly, so what binding energies tells, basically binding energies tell how strongly that inhibitor binds to the microbial or human component which we want to block. Okay, so there is these are a lot of calculations and means mathematics of this binding. So anyway, so basic binding mechanism could be of several types. The, it depends upon what kind of interaction between the drug and the microbial or host component which we are going to find is happening. So what kind of features it has, like complementarities, like lock and key has complementarity. That's why the key goes inside the lock. Okay. So the shape of the ligand or its physicochemical properties, like what kind of charge it carries, the binding pocket on the target and the charge on the ligand or inhibitor, and also their shapes. Okay. Okay, so this that's how it works. Basically, we ask computer program to one by one fit in that inhibitors, means potential inhibitors or potential molecules inside the that component which we want to block, that target. So it will put inside, and then it will calculate whether the binding is strong or not, and then one by one. So like we do like 10 lakh molecules, we screen 10 lakh molecules against one single component in like 4 or 5 days, and then it depends upon how faster computer you are using. So now people are using supercomputer or quantum computer, that will be very fast, like that will be in few hours. 10 lakh molecule library can be screened. Okay, so this kind of analysis can be done. And then another type of computational analysis also we do, like we check if the molecule will go inside human body, what is its fate. Okay, so two things are there. First, the molecule should reach to the particular organ where the infection or where the disease is happening. Okay, so it is called as bioavailability. It should reach there and should be available so that it can bind to the target component where we are means we are targeting, like micro, if it is a microbial component. So let's say infection is in our lung, so it should be the molecule which we take orally. It should first go metabolize in our stomach, and then it should assimilate, it should absorb by the blood, and it should reach the lung, then only it will work, na? So that kind of information we can computationally analyze, like the molecule, what is the rate of liberation of that molecule, or absorption of that molecule, or metabolism of that molecule, or how it will be excreted. Because the drug molecule is, when it is taken, it is like a prodrug, and then it will, when it will go in our stomach, it will be metabolized, it will be converted into some other molecule, in some other form, which should be active form, which should go and which should treat the disease. Okay, so these all are analyzed, that when it will go to human body, then what will happen. Similarly, we analyze like whether it can cross, it will cross the blood brain barrier, because you know, if molecules will cross blood brain barrier, it can reach to our nervous system, or it can reach to our brain, so it can cause toxicity. And brain is so important that if there will be toxicity in brain, then whole body will stop functioning. So it is very important. That's why, so we check whether the molecule has potential to cross blood brain barrier, BBB. So it should not be, if we are making a drug for lung infection, it should not cross blood brain barrier. Okay, similarly, but if we are targeting a molecule, we are going to make a drug for nervous system, then it should cross blood brain barrier. Similarly, we also analyze whether this molecule this molecule by any chance could cause carcinogenicity, or this molecule can cause cancer. If that also we can analyze, analyze in means before the computational techniques. So that we will all do that kind of analysis, and in the end, then we proceed from various analysis. We proceed to those molecules which are best for our consumption, for our purpose. So there are many drugs which were discussed in context of COVID-19. Like, but you see, like that's why I said in popular culture and media. But you see, science is not, science don't go like overnight. It is a continuous process. And because you see, we need to check many things. We need to check a lot of things, and we need to be very careful. If we will not be careful, then what will happen? It will be dangerous. It can cause problem. But media, like during the pandemic, there is pandemic of fake news also. So for example, people were across the globe, it was happening. People were like somebody proposed chloroquinone and hydroquinone, which is a quinolone derivative, which is basically anti-malarial and anti-rheumatoid arthritis drugs were there. So later on, it was found out that it was found out that it is not functioning against COVID infection. Similarly, remdesivir, similarly other drugs like favipiravir. So there are a lot of drugs which was making the headlines, but later on, when people have analyzed it in a bigger setting, or when trials happened, it was failed. So like ivermectin. So our government was also giving precautionary dose of ivermectin to medical practitioners. So there was a lot of data, and means maybe more specialized for more specialized people. If they are interested, you should go and you should see the mechanistic information now available, that it is not of any use basically, or in some cases even this kind of drugs were doing more damage uh, than helping. So for example, this was also highly touted, this 2DG, that is a modified glucose molecule, modified D glucose molecule into deoxy glucose. That was also, that this drug was also touted, and it is basically anti-cancer drug, and it was announced by our defense minister, because some DRDO scientists, they have reported some preliminary information about it, that this anti-cancer drug can work against COVID-19. But after that, now nobody knows where it is. But media will pick, media will flash, misleading headlines. But science don't go like that. Science has a process. It goes with its own speed, and it will do all necessary precautionary testing, and then only it will release the data. And the good thing is, in science, it is not like something is written in the book written by God. So it is challengeable always, and then like this drug, nobody will know. If it is not worth, then nobody will talk about it later. Okay, but media will pick, and media will do big scene out of it. Like Patanjali has what Patanjali Ayurveda was selling coronal kit, and they were claiming that it can treat this SARS coronavirus infection. Means there are, and there was a big TV shows on that, but later on our government's medical agency ICMR has given a precautionary note and press release, and also there were a lot of instruction from Ayush Ministry that you should not make such claims. Okay, so similarly there was also like dexamethasone or cartisone or steroids, which were okay. So steroid help in all means
Excuse
it
me,
is like
sir.
a
Excuse me. Sorry
yeah.
to interrupt you, sir. Actually, we will have to start our next session. That's it. Please
Okay. So this
continue
is
your.
this is last
Thank you. Sorry,
slide.
sorry.
Okay, so with this, I want to thank the organizers, and this is my group at Central University of Himachal Pradesh, and later on I have been 5 years back I shifted to Lucknow, Baba Saheb Bhimrao Medical University, and I am also thankful to my institutions and Government of India agencies who basically fund, and with the help of them I do my research with my students and colleagues and collaborators. Thank you very much, and with this I stop my presentation here. If you have any question, please feel free. Thank you very much.
If anybody has question, yeah, sir, there is a question, sir. Excuse.
Okay.
Good afternoon, sir. Thank you very much. The session was very informative and very elaborative. Sir, I just want to ask that we are listening, hearing a lot of cases of heart cardiac arrest after COVID.
Correct.
So, is there any side effect of COVID vaccine due to which it is increasing?
Yes,
And you have
so let me explain. Yeah, let me like I said, science is when science give answers, it give in very pragmatic way, and it is very it remain very cautious. So like vaccine means how vaccines works. If you look at it, so when you take vaccine in vaccine, what you take either that attenuated or dead microorganism, or you take a component of that microorganism, and what happens, you generate an immunological memory inside your body, in your immune system, so that next time, okay, that is a either dead microbe, so it will not create disease, or part of the microbe which will not create the disease, but it will generate an immunological memory in your body, and next time when the real virus or real bacteria comes, then what happens, then basically your body will respond because it is now trained. Okay, so it means we are inducing immune system, and immune system, how immune system works, it is called as, okay, if I will say it will it will induce a process which is called as inflammation. Inflammation means your cells, immune cells will divide like in normal course. If there is any infection, you will see inflammation and pain. Why this is? Because the immune cells
body
which
reacts.
are going to fight, those cells will divide, and they will go to that side where the infection happens. So inflammatory response is must. So what happens, so similarly what is observed, that this vaccine causes an inflammation, obviously, because it is necessary for the infection, for its function. So there is a specific inflammation that is called as myocarditis. Means
Yes.
heart muscles, heart tissue is inflamed. Okay, so but what is seen, that in most of the cases, like in 99.8% cases, only in this inflammation is recovered within few days, like within few months, you can say complete inflammation was gone within few months. But in some cases it may happen where there is history of cardiovascular disease, or there is genetic makeup of cardiovascular disease. But what is reported by so on by now is like in 8 lakh people, 8 lakh people, there is one case which has faced this thing. Now there is a barter, means whether you want to die by infection or you want to take this risk.
Yes.
So in younger people, for example, there is a very less chance that COVID can do any harm. So in younger people, the other thing is the inflammatory response is very strong. So basically for younger people, that's why the European Union has recommended that for younger people, if they have some history or some weakness, or some means they are prone to infection, then only give the booster dose. Otherwise you don't give booster dose. But in the USA, they have, so in on this topic, the scientific community and clinicians are divided, and government of India still have to make some decision. But like for older people, they have given that booster dose should be given.
Means booster dose is more risky.
Booster dose is more risky because the myocarditis means what people who are immunologists and others. Actually, I wrote a piece in Hindi. If you search, you will find it, or maybe I will send it to organizer, the person whom I'm connected with this conference.
Okay.
So that is everything is written. So the same concept I have written in that.
Okay.
In a new, yeah, in an editorial, so you can read that. So
Okay,
so older people are, yeah, so that's why for young people, so it is a barter, means whether if you are more prone for infection or you are more prone for myocarditis. So myocarditis is also recovered in most of the cases, but if the inflammation is seen in the heart tissue, so it means it is doing something.
But nowadays we are hearing a lot of cases of cardiac arrest during
Young people,
or during morning walk.
Yeah,
Yes.
that is
And there are few reports which are indicating that this mRNA vaccine.
Definitely. So this mRNA vaccine was found more means more linked to myocarditis.
Although with the development of vaccine, we could control this pandemic. This was a big achievement. Yes.
Yeah, this is big achievement. So always
there
it is a barter.
There may be some,
it is a barter.
Yeah. Thank you so much.
Okay. Thank you very much.
Any other question? Thank you very much, sir, for such informative session which focused on the recent trends and components of development in the structure-based drug design using advanced molecular modeling, and how technology plays an important role in this process. It will definitely help us in future. Now it's time for the high tea. Request to all of you kindly assemble in auditorium after 10 minutes. Thank you very much, sir.
Thank you very much.